Okay, let's start now. Welcome everyone to the Greener Hub webinar series. My name is Evelyn Zuniga, and I am one of the communications officers for the Greener Hub project. For those who are new in the webinar series, the Greener Hub project officially started in September 2022 as a coordination and support action of the European Commission under the Horizon Europe program, and it currently represents 15 ERANETs, co-funds, and self-sustained initiatives in the field of agri-food and biotechnology. We started with these breakfast clubs last year in October with the topic of circularity in agriculture. So if you're interested in topics related to mixed farming systems, mitigation of greenhouse gas emissions, sustainable farming, and circular food systems, all our recordings are now available in YouTube. Now and for these 3 months since March until now, since January until now, we focus on a different topic, which is biotechnology to advance sustainable solutions for bioeconomy, where we had topics related to plastic degradation, production of chemicals in a sustainable way, biofuel production, protein engineering, among other topics. Just a few housekeeping announcements. Please feel free to put your questions in the Q&A and chat section that I have activated both of them. And I'm going to address as many as possible with the time that we have, which is 1 hour. And if at the end of the session you have a question for either of the speakers, please address the speaker in the question. Today we have two panelists, Professor Bettina Sievers and Professor Trygve Brautaset. We're going to open our round of presentations with Professor Sievers, and give me 1 minute that I'm going to share one screen. Okay. Professor Bettina Sievers is professor at the University of Duisburg-Essen in Germany and is head of the group of molecular enzyme technology and biochemistry. Her research interests are focused towards the central carbohydrate metabolism of hyperthermophilic and thermoacidophilic archaeal organisms, exploring the utilization of extremozymes for biocatalysis and process optimization in bioeconomy. Her team uses a combination of classical biochemical and molecular genetic approaches together with modern and high-throughput technologies in functional genomics and systems biology, as well as metabolic engineering and synthetic biology approaches. Her talk today is entitled Thermophilic Bacterial and Archaeal Chassis for Extremolyte Production, which is part of the Hot Solute project from Eraco Biotech. Professor Bettina, thank you very much for joining us today and for sharing all the insights on your project. And I'm just gonna stop sharing my screen and you can start sharing yours, and the audience is all yours. Thank you very much.
Thank you. Okay, so I think you see my screen now. So
yes, it's in presentation now.
So thanks again for the nice introduction. And this project Hot Solute is actually in the past. So we started 2018 and it ended 2022. But I'm happy to present what we achieved in the project, and maybe also what's always my intention, to get you a feeling about the excitement about thermophilic organisms, as you see already in this picture. So to start with the partner, so I was the coordinator of the project in Exeter. We had Jennifer Littlechild and Niklas Hamers. They are expert in thermophilic bacteria. We had Daniela Monti in Italy. She is actually an expert in biocatalysis. Felix Müller joined from Evonik Industries, so one industrial partner. And Elisaveta Bonch of Smolowskaya is from the Vinogradsky Institute in Moscow, and she's into metagenomics and was a source of novel enzymes. And we also included modeling with Jackie Snoop from Stellenbosch. And as you see here, this was the central idea we had. So actually, we wanted to make hot solutes. These are extremolytes. I explain later what they are. And they can be either made by enzyme cascades. That was one basic idea. And the other was to use thermophilic cell factories. And here we use the expertise of the Exeter group with Thermus thermophilus. It's a bacterium that grows at 70 degree pH 7.5 to 0.8. And our expertise is on the Archaeonzophilobus acidocaldarius, so 75 degrees to 80 degrees and pH 2 to 3. So here we have thermoacidophilic archaean as small model organism. Then the idea was also to look for novel enzymes in thermophilic environments. And as I said, to use either an in vitro cascade where we use the enzyme from thermophiles, or in vivo where we use these two novel chassis for the production of extremolytes. And we also included modeling, and the idea was then to scale up the production and to further analysis, product testing, and so on, together with our industrial partner. And just because I'm working on thermophiles for quite some time, I have always a feeling I have you to share my excitement about this organism. So if you think about what we are doing today, we often rely on certain model organisms, so E. coli, bacillus, yeast, and we still there, we don't really understand the organisms. We have up to 30% hypotheticals, but there are many organisms beside of this, so that we have never really used the biological diversity that's around regarding organism, but also metabolism. And we talk here about the metabolic dark matter. And I think this is nicely shown in the picture. You think you know everything by this yellow dot, but you have a totally different situation looking at different organisms, different enzymes, different pathways. And this is something I think we have to use for biotechnology. And just to share some ideas what I think high temperature can help. So you have often, if you think about lignocellulose degradation, you need the high temperature because substrate solubility is increased, and it makes the substrate more accessible for degradation, easy mixing for cultivations. The thermostable enzymes, so-called extremozymes, are exciting because they are so stable, they even work in organic solutes. Then you can also reduce the energy for input. You don't have to think about microbial contamination if you grow Zophilobus, so there is no need for antibiotics. And you can also easily separate volatile products just by distillation and during the fermentation. And all this, of course, allows you to think about novel process design and about novel one-pot strategies. And just also since archaea are for me, of course, my central research subject for many years now, I would like to share you also the fascinating topic of archaea. So I think you are all aware that we now discuss this two-domain tree of life where we have like bacteria and eukarya and archaea. These four groups here. And the idea is now that actually the eukaryotes, so we are actually were emerging from an archaeal domain of life, so that this is now the novel two-domain tree topology we discuss in literature. And what's so fascinating about archaea is that they have an archaeal mosaic character. So somehow they are bacteria-like. Their size and forms are similar, but if you look at the genetic organization, they are also like bacteria. You have operon structure, circular chromosome, plasmids in the cell. But if you look at the information processing, like transcription, translation, this is more like eukarya. So you have a typical polymerase II promoter, Tata box, preside, and also post-translational modifications, phosphorylation, glucosylation is more eukarya-like. But there are many really archaeal-specific topics, and this is really the unique metabolic pathway. So we find a lot of novel pathways, novel enzymes, even in the basic metabolic pathways. They have a unique membrane lipid composition. They don't have fatty acids. They use isoprene chains, ether linked to glycerol 1-phosphate, and also the cell envelope. We never find murein. Often they just have an S layer, pseudomurein, or they don't have an envelope at all. So these are really unique, and all this novel metabolic diversity makes them really an interesting topic to think about. And our model organism we use in the lab is Zophilobus acidocaldarius, a member of this TAC superphylum, and it was originally described by Brock. It's an aerobic heterotroph, so it's quite easy to grow. We just need shakers we can heat. And of course, we have to see it's a thermoacidophile, so pH 2 to 3. The genome is deciphered, and what's most important for us is that it's really genetic tractable. That means we can do knockouts, we can do protein expression. So it's a host where we really can do metabolic engineering. And for the project, now I come more to the project part, it's of course the question, what are compatible solutes? So compatible solutes are found in all domains of life, and this is really closely related to the response to environmental stress like heat, desiccation, soil stress, and compatible solutes actually help the cell to respond to the stress conditions. So these are typically organic low molecular weight compounds. They are highly soluble, can be accumulated in very high concentrations in the molar range. And what's important here, it's where also the name comes from, compatible, because they don't interfere with the central metabolism. And their role is then really to protect the membrane, proteins, DNA, and RNA from these stress conditions. And this is what typically happens in a cell. So either upon stress, compatible solutes can be taken up, or they are synthesized in the cell, and then they help to protect the structures. And if you have a stress relief, typically the cells use often mechanosensitive channels that open. For example, if you have, that's well known from E. coli, if you have salt stress, actually E. coli accumulates trehalose, and if you have the stress relief, the osmotic downshift, then actually they just open mechanosensitive channels and throw out the compatible solute that can be later on be degraded. So in this case, trehalose by trehalase, and the carbohydrate can then be taken up again. And typically, this is a quite diverse family. So in many mesophilic organisms, yeast, plants, E. coli, as I said, we find sugars like trehalose, sucrose. In other organisms, often halophilic fungi, we find polyols, glycerol, arabitol, inositol, and also, for example, this resurrection plant is quite famous that prolongs long time of without water, and you can then really actually deactivate the plant again. And what's also well known are these amino acids and derivatives. Many of you maybe heard of ectoine, hydroxyectoine. They came often from halophilic or halotolerant organisms. This is a picture here. So the question is, what are extremolites? So extremolites is the names for solutes we find actually in extremophiles, and I just show you some of them here. So it's, for example, cyclic diphosphoglycerate shown here. We find in many hyperthermophilic methanogenous species. We find monocyclocerate. This is a more common solute in many organisms, and also quite special for hyperthermophiles like thermococcales or methanogens and archoglobus is dimyoinositol phosphate. And the important thing for our project was here that the pathways are known, so the enzymes that do the job have been known, but actually there was, so far, it was really hard to produce at least these two compounds in current platform strains like E. coli. So this was then the basic idea, and of course, it's important, so where can we apply these products? And for example, for monocyclocerate, it is reported that's actually active in disease and health in disease. So it has been shown to have an effect in Parkinson, Alzheimer, Huntington disease. Then, of course, if you think about cryopreservation or stabilization of biomolecules, if you buy your enzymes from a company, typically these are stabilized, or your food. If you think about green peas, typically there is trehalose added to keep the color of the peas. So this is a quite common thing that compatible solutes are used. And of course, in the cosmetic industry, many lotions, sun creams, and especially anti-aging products actually harbor all these compounds. And also, as I said, in the food industry, this is commonly used. So there's a very broad application. And of course, our idea was to provide this novel extremolites would be very helpful actually to provide new compounds here. And I just picked some examples to show you the progress we made in the project. So one compound we went for was the cyclic 2,3-D phosphoglycerate, and it was mainly Christina Stracke who worked in our project, and she's still working on this. And what we actually did is first, as I said, the two enzymes involved has been known. So what we start with is 2-phosphoglycerate. Then we have a 2-phosphoglycerate kinase that phosphorylates to 2,3-D-phosphoglycerate. And then we have a cyclic dephosphoglycerate synthase that actually makes a ring closure here, so that you end up with a cyclic 2,3-D-phosphoglycerate. So as I said, this pathway was known from hyperthermophilic archaea, especially methanogens. And what we did here, we first started to go for an enzyme cascade. So what we did, we achieved the expression of the proteins, as you see here in E. coli. And this is something I would like to point your attention to. That's also perfect working with our thermophilic proteins. If you express them in E. coli, you often can just do a heat precipitation where you then nearly get a pure protein fraction afterwards. And then we always provided the protein to our partner in Exeter. They are also experts in structures solution, and they also were actually able to solve the structure of this protein, the cyclic dephosphoglycerate synthetase, and this was actually a novel fold and a novel protein family. Okay, so how did we continue then? So first we characterized the cyclic dephosphoglycerate synthetase, and again, this is the main reaction, and for this we used actually a continuous assay, and you see we use 55 degrees because then you can work with available mesophilic auxiliary enzymes. So the idea is that to actually monitor here the ADP release using the pyruvate kinase and lactate dehydrogenase, just the yeast enzymes you can buy with Sigma Aldrich, and then you can just monitor the NADH consumption. And this is what we use for the characterization for both the substrates. And as you see, the enzyme has a quite nice or high maximal velocity with 21 units per milligram, and also the affinity of the enzyme to the substrate was quite nice, so in the millimolar range. And what we quite early observed is that actually we get a full conversion of the 2,3-D-phosphoglycerate, like shown in this plot here. So we were able to monitor this 2-phosphoglycerate release and then the ADP release here. So what we observed here, 1 millimolar can be nicely converted. And then Christina looked for other technology and she established phospho-NMR in the group. And what you see here with 1 millimolar, this is a time curve, as you see here, up to 40 minutes. And what you see, if you just look at the 2,3-D-phosphoglycerate and the cyclic dephosphoglycerate, what we see here after 16 minutes, the 2,3-D-phosphoglycerate is consumed, and you see that the CDPG is formed. And then we always tried higher concentration of this, and you see here we can also get a full conversion of 100 millimolar. And the nice thing is we can do this really with the protein, just doing E. coli expression, heat precipitation. We get the nearly pure protein that does this conversion at high temperature. And we used also Jackie helped us to model the consumption. So what we see here is again the consumption of 2,3-DPG and then the ADP release. These are the modeled open points, and these the full points are actually the real measurements. And what you see that then the model nicely overlaps with what we observed in vitro. So what you also see that, of course, the incubation time is extended, but at least you can just incubate the enzyme with the substrate over time and you get full product conversion. So this is a very nice enzyme cascade, and our partners in Exeter then also use Extremophilus for the whole cell catalysis, and they were also use the enzymes from Methanothermus fervidus, and they could then also show that over time thermus actually produces 2,3 cyclic phosphoglycerate, and they were actually able to observe 650 micromolar of CDPG in these cell extracts. Okay, as next, I would just briefly go into another topic, and this is the production of monosylglycerate. I said at the beginning that this is a compatible solute that's more spread, so we find it in all 3 domains of life. And what's quite interesting for us, there are also many extremophiles around that actually use this compatible solutes. And in Rhodothermus marinus, 2 pathways have been identified and published by Helena Santos group. And what they start with is activated mannose, so GDP mannose. And then you can have either a manosyl 3-phosphoglycerate synthase that uses 3-phosphoglycerate to make manosyl 3-phosphoglycerate, and then a phosphatase to make monosylglycerate. Or you can also do this in one step with the monosylglycerate synthetase by using glycerate as co-substrate. And as I said, this is a quite interesting solute because it has been shown to be actually active against Alzheimer's disease. And what we did, we tried expression of different enzymes in our model organism, Zophilogus acidocaldarius, and we were able to show that the enzymes from Rhodothermus, this is the one-step enzymes, as both enzymes of the 2 steps are actually produced. The enzymes are nicely active, also produced from in Zophilogus, and this is also what has been published from the Santos group before on the enzyme from Rhodothermus marinus, so that fits quite well. And then we actually looked for the production, and what we observed is that using our Zophilogus strain, we were now able to see actually production, and you see here the growth curve, and what you see here's a specific activity, and what you see that most of the monosylglycerate is then actually found in the cell, in the supernatant, so that it can be easily purified from the supernatant. And the concentrations are not so high, so that's something we currently work on in the lab. And this is already what I would like to present here today. So what I think what we really nicely succeeded in the project was that we actually provided in my group an in vitro one-step synthesis for cyclic phosphoglycerate by the enzyme from Methanothermus fervidus. And what we are currently doing in the lab is that we actually try to extend this enzyme cascade to come to cheaper substrates we can use, and also introducing an energy recycling enzymes in Zophilogus acidocaldarius for whole cell catalysis. We established the monosylglycerate production, and here we actually try now to actually improve the strain by engineering the UDP mannose synthesis pathway to improve this, and by also including the 2-step pathway and also enzymes from other organisms. And finally, we also start now to work more on the demyoinositol phosphate synthesis. This is also a very interesting compound that can't be or can be synthesized by other mesophilic platform strains so far. Okay, and this I think, as I said at the beginning, what's the important thing for me, we have all this biological diversity around, and I think novel platform strains and novel enzymes really can have a major role in biotechnology. And this can be even further extended by protein engineering so that we later on can modify the enzymes. And I think this can be really a big player in our aim to reach the sustainable development goals that actually we have. And with this, I would like to thank everybody who was involved in the project. And before, I still have an announcement. So we have just Marie Curie network that started, and I'm looking for interested PhD positions, also the other groups. So in my labs, this must be a non-German student that can apply, but in the other groups, there is also a lot of things going on. We are interested in N-glucosylation, so glucosyl transferases, also with a biotechnological focus. And as you see, Essen is close in the center, in the heart of Europe, as we always say. And I think I also have a quite nice group, and we would be happy to welcome new students in our colorful university here. And with this, I would like to thank, of course, all partners that were involved in the Hot Solute project. So I focus now mainly to what we did in the laboratory, but I think it was really a wonderful collaboration, and I really thank the funders for the possibilities to get this started, because I think this is, of course, a bit always outside the box, because not many people work with thermophiles, but I think for us it was really very important to get started on this, and also the metabolic engineering, and to get more into biotechnology with these organisms. Yes, thank you very much.
Thank you very much, Professor Sievers. I just want to remind people that the Q&A section and the chat section are open for you to type your questions. And let me see. There's a question here. It says, what are the native compatible solutes of S. acidocaldarius? Um, would it be possible to overproduce them with its native enzymes?
Mhm. That's a wonderful question because I didn't have time, so I didn't mention this. But actually, Zophilobus makes trehalose as compatible solute. And one project we also followed up was actually to unravel the pathways, because again, here we have a lot of novel enzymes. So 3 different pathways. Trehalose can be done from glucose, glycogen, or glucose 6-phosphate. And we unraveled the pathway by doing the knockout strain, so it's accumulates in response to salt stress. That's what we were able to show. And actually, we used the triple knockout of the 3 pathways for the production of monosylglycerate. So this is also a basis we did for the production of the compatible solute. But there is, of course, also for trehalose production, this is a quite interesting strain, and you can actually isolate trehalose also from Zophilobus. Yeah.
Okay. I do have a question here. So did you have any problems, or what are the bottlenecks on like upscaling a system like this for like bigger, or this
Mm.
is like already in
Yeah.
big tanks?
So actually, what we are still fighting is that, of course, many companies have their established systems. And of course, if you change everything to high temperature, that also means that you have to change the established systems. And therefore, we also collaborate quite closely together with a group at the Technical University in Vienna, Oliver Spadut, and they are actually bioprocess techniques. So we actually were able to get really high cell densities using a continuous fermentation cell retention system. And there we could reach now ODs up to 60. So this is now a scale where it really gets reasonable. And I think this is also important, of course. And we still have to improve the genetics. So we just established CRISPR-Cas, but still working with this not state-of-the-art organism. It's not E. coli. It takes longer to do the knockout mutants, for example, and also to integrate things into the genome. And we still have to improve these methods.
Hmm. Okay. There is somebody asking, do you have any idea how energy efficient this would be, as the bacteria are thermophilic and need to be in and need the heat?
Actually, they need the heat for growth. So you, as I said, typically we use 75 degrees for growth, and at 50 degrees you have hardly any growth of these organisms. But this is also something you nicely can play with around for if you think about product production, of course. Also something you have to realize that some products you might want to make are not thermostable, right? So then you can think about doing things like using cold shock inducible promoters, which we have in hand, to then do a temperature shift and then to use the enzymes available for product formation. So this has been also already done in the field for pyrococcus. So I think there is something still to play with. And regarding, of course, you have the input heat you need for cultivation. But as I said, so typically if you use E. coli fermentation, a lot of energy is required because during metabolism, the organisms produce heat, and then you have to cool down actually the fermentation. So you also need energy here. So I think then it's somehow, I never calculated this, but this is of course something we want to consider in the bioprocess design now.
Mm-hmm. We had another question here. It says, are there any extremolites known or extracted from Asgard archaea or not yet?
Actually, I have to admit that I didn't check carefully the genomes of Asgard archaea, so I'm sure, as I said, you find compatible solutes everywhere, so yeast, whatever you look at, but I never checked what pathways are commonly found there.
Okay. Um, so you mentioned that you are testing this with you're trying to do Cas9. Are you working on with Cas9 right now? So there are enzymes that are stable to do Cas9.
Mhm.
Yeah, the modifications at high temperatures already.
Yeah, yeah. So has been reported for Zophilobus islandicus. There is this system established, and we just published this for Zophilobus acidocardaris.
Okay. And so
But
when
sorry, it's not the Cas9 system. We have an archaeal specific CRISPR system we are using, but this works similar.
okay, so you use
Mhm.
okay, I understand.
Mhm.
So when you isolate, maybe I'm misunderstanding, but when you isolate the enzymes, and then so you have to keep them in like in hot temperatures or low pH, otherwise they will denaturalize.
No, actually, it's what you have to think about extremophilic enzymes. So, as I said, they are very stable. So, the protein to be active at high temperature, you all know if you like cook an egg, your protein denaturates, right? And you have your cooked egg finally. But this also is a challenge for protein. So, somehow they have to be stabilized, and that means they are more rigid. And they need the temperature actually then to do the conformational changes you need for being an active enzyme. And there are several tricks that have been reported how protein achieves this, but this also has for many or most enzymes the saying from hyperthermophiles, they are nicely active at 70, 80 degrees, and you can go low to 50 degrees, then you still see activity, but that's only a part of that of at 80 degrees. And if you go to 30 degrees, many of these enzymes are just not active or only have remote activity. So the enzymes are really adapted to this temperature range of the organisms.
Yeah, perfect. That was very interesting. So I guess we can move on. I don't see more questions in the
Yeah.
chat. Oh wait, hold on. There's another one. Have you thought about other possible model organisms like yeast to test the production of such industrially relevant compounds?
Mhm. Actually, this was a bit our starting point because during my PhD in the group of Reinhard Hensel, we also already started to try the CDPG production in E. coli. And that's why we realized, I don't know how many PhD students worked on that, but really like 3 students, and we really end up with minor amounts. So we realized this is not the way to go.
Mm.
And therefore we said, okay, for using these enzymes, we really need the thermophilic expression host. And this was actually the main idea of this proposal, that we said, okay, some things you just can't produce in E. coli.
Okay. Yes, thank you very much. Thank you everyone for your questions as well. And I think we need to move on to our next presentation. Professor Trygve Brautaset is currently a professor of synthetic biology at the Department of Biology and Food Science, and it's currently a professor of synthetic biology at the department, a leader of the Center for Digital Life in Norway at the Norwegian University of Science and Technology. His work focused on microbial molecular biology and microbial cell factories seek to replace sugar-based feedstocks with single carbon compounds, paving the way for sustainable production of valuable chemicals. His team uses cutting-edge techniques, blending systems and synthetic biology approaches to engineer methylotrophic organisms tailored to diverse environmental conditions and metabolic requirements. And today, Professor Brautset's talk is entitled Replacing Food Competing Feedstocks with Methanol, CO2, and Methylamine for a Sustainable Bioeconomy, which is part of the MCM4SB project from ERACO Biotech.
Yes, okay. So thank you very much. Thank you for this invitation to present about our project. So yeah, replacing food competing feedstocks with one carbon compounds for sustainable bioeconomy. So yes, the reason I'm here, I'm leading one of these projects, but what I will try to do is to put a little bit what we do in this project into context. So you can go to the next slide. I will start relatively basic here. But as many of you know, industrial biotechnology.
Sorry. Yes, I'm sorry. Yeah.
Yeah, so industrial biotechnology use microbial cell factories. They use and they develop microbial cell factories for sustainable production of value-added products. And microorganisms can be trained to produce a very wide variety of different products for different uses. And if you go to the next slide now, but in line with this, there is sort of an increasing concern which has to do with the raw material that they feed the microorganisms. So my very simple illustration here shows this. So you have the bacterium, which is the cell factory. It needs a carbon source to grow, and then the metabolism it does can then be kind of manipulated so that you can have production of particular compounds that you are interested in. And traditionally, and also today, raw materials are sugars and molasses. And this gives an unfortunate kind of competition with the food sector because these rely on use of cultivable land. So industrial biotechnology needs alternative raw materials. And this is particularly important when we talk about products that are produced in high scale. Next. And that is where one carbon compounds might come in as one alternative as a feedstock. And when we talk about one carbon compounds, I think most typically it's methanol, methane, and carbon dioxide, but there are also others that could be there. They are non-food. When it comes to price and availability, there is a lot of things happening with regard to, in particular from methanol, production facilities. So it's really a source that is where the production scale is increased globally today. And also they are completely consumed by microbial fermentations. So then you can go to next. And this is where methylotrophic bacteria comes in, because there is a special class of bacteria, the methylotrophs, and they have this innate property that they can consume and use one carbon compounds as sole carbon and energy source for growth. Next. And in our group, we have worked with one particular methylotrophic bacterium named Bacillus methanolicus. And this one is one model organism in the project that we are currently running. This is a gram-positive bacterium, and also a little bit similar with Bertina. It is a thermophile, but I would like to call it a moderate thermophilic bacterium because the growth optimum is around 50 degrees. And if you go to like mesophilic temperatures, it can hardly grow at 37 or something. So it is a thermophile, but it grows as at moderate high temperatures. And this is actually also a property that is novel with respect to using this organism. Over the years, we have developed omics technologies, genome scale models, developed genetic toolboxes, so that more and more it becomes a model organism that can be used to try to develop cell factories that can actually convert methanol into different products. And there are also wild-type strains and classical mutant strains that overproduce the amino acids glutamate and lysine that sort of shows the capacity of this organism. Next. So what I aim to do here is to show you a few examples. I will start with one example that was a little bit beyond the MCM4SB project, but the idea for me is here is a little bit to try to illustrate how we work, what are the challenges that we meet, and also how we try to handle them. And acetylene is a commercial product that has many applications in pharmacy. And we wanted to try to make a cell factory out of Bacillus methanolicus to convert methanol into acetylene. This is a non-natural product for Bacillus methanolicus. So here we have to construct what we call a synthetic pathway, and you can see it here on the figure on the left side. The precursor molecule is pyruvate. And then we introduced two heterologous genes, acetolactate synthase and acetolactate decarboxylase, that created a pathway where pyruvate can be converted into acetylene. And this resulted, in short, I can say, into a titer of 0.26 gram per liter, so which is a kind of a good proof of principle starting point. And then to illustrate what is typically done after you have constructed a biosynthetic pathway that works is to look into the supply of the precursor molecule, which is in this case is pyruvate. And then you can take the next slide. And here I have a little bit changed the metabolism that is relevant, because now we look more into the central metabolism of the cells. And pyruvate can be derived from the TCA cyclus, and there are anaplerotic routes. Some of them are illustrated on this not so very nice illustration, I must say. But if you click on the next slide, I will illustrate that there were two genes that we targeted. We tested many different of them. And the idea here was, can we disturb this metabolism to have a higher supply of the precursor molecule pyruvate? And what we found was that overexpression of malic enzyme and isocitrate lyase resulted in a doubling in the titer of acetylene to 0.42 grams per liter. So the idea about this is to show that when you work with metabolic engineering, sometimes you have to have a focused perspective to construct a pathway, a biosynthetic pathway that is functional in the bacterium that you work with. And then when this is optimized, typically the next phase is to look into the central metabolism to increase the precursor supply. And this also leads us then to our project that is currently running. MCM4SB is a horrible short name, but this is a short name for replacing food-competing feedstocks with methanol, carbon dioxide, and methylamine for sustainable bioeconomy. And the figure here illustrates our project. I will come a little bit back to that, but also here you can see our consortium. One of the nice things I think about ERA co-biotech projects is that you can really make this international consortia of groups that are not too large, but sufficiently large that you really can put together very good teams. And many of the partners in these projects have collaborated on previous ERA co-biotech projects. So next slide. And this is to illustrate what is kind of the goal here. We target, in principle, 3 different one carbon sources, which is methanol, of course, but also carbon dioxide and also methylamine. And you can see them on this figure. They are labeled in yellow color, so they come into various places. And the products that we target are native compounds found in Bacillus methanolicus and also in the other organisms we are working with. It's malate and methyl glutamate, and you can see them boxed in here. One is a bulk chemical produced in very high quantities. The other is a specialty chemical which has a high price and not necessarily produced in the similar high quantities. And in this project, we have two model organisms. One is Bacillus methanolicus, I just mentioned, and the other is some Methylobacillus strain, which has a certain code for the moment, because this is one that was picked by our industry partner, ACS Bio, and therefore the full name is not given yet. Next. Also here, I will show some examples of what we have been doing and what we are doing also here a little bit to illustrate the way that we work. So this figure on the right side is showing kind of an illustration of main metabolic pathways. Carbon dioxide is here, methanol is here, and one precursor molecule that is important if you want to produce malate is oxaloacetic acid in the middle label there. And the purpose of what we've done here is to see if we can increase the supply of oxaloacetate in the cells. And we had two target reactions. One is by PEP carboxylase, which on this figure goes from PEP to oxaloacetate, which, yeah, you can see it there. No, on the left turn there on the top. And this is a non-native reaction in our organism. And then it is from pyruvate to oxaloacetate, which is a native reaction. And then if you go to the next slide, which is a relatively busy slide, but what I want to show you here is that when you work with a thermophilic bacterium, which also has a very high AT content in its genome, when you want to recruit enzymes for a purpose like here, we usually have to go broad. We have to go to many different organisms to recruit the same enzyme. So you see PEP carboxylase, there are five different variants from different organisms, and then pyruvate carboxylase is the same. As Martina said, when you work with thermophilic organisms, you typically try to recruit genes from other thermophilic organisms. But this does not always work for us, because many of them, they have a temperature optimum that is too high. So at 50 degrees, they don't work. But what we also see is that very often, surprisingly, I would say, it works very well also from mesophilic bacteria. Many times we have very functional enzymes recruited from Escherichia coli, for example, that works very well in our bacterium. So that was, this is an illustration of this. And if you then go to the next slide, here we have 10 strains. And what we typically do here is to measure enzyme activity, and then also as an indication of how well they can cause increased supply of oxaloacetate. We here measure this indirectly by looking at lysine production in the cell. So if you look at the left panel top here, you can see that you have different PEP carboxylases, and the enzyme activity in vivo in methanolicus is very different. At the bottom here, you have one that is very active. And then if you go to the bottom panel, you can see that it also causes increased production of lysine. So this is a very good indication that this particular heterologous gene will cause increased supply of oxaloacetate in Bacillus methanolicus. And then we use this together with engineering of the biosynthetic pathway in question that relies on oxaloacetic acid. Next. Yeah, and then as I said, this project has two different model organisms. The other one is Methylobacillus, and this is mainly work that has been initiated by our industry partner, ACS Bio in Slovenia. So to explain a little bit here we have a different C1 compound, methylamine, because this bacterium can use methylamine as a carbon and energy source. And the product we target here is N-methyl glutamate. And in principle, I think the figure on this slide shows very easily what is explained in the text on the left side. So methylamine is in the center top there, and it has two different catabolic routes. And one for us, the one that goes via methylamine glutamate methyltransferase is the one that we want, because this leads to our product methylglutamate. So what we do is that we downregulate methylamine dehydrogenase with the red cross to the left to try to channel the carbon flow towards our product. And then we also knock out methylglutamate dehydrogenase to prevent that our product is converted into glutamate. And these genetic modifications, we achieved 0.35 grams per liter of our product methylglutamate from methylamine. And this is, when you compare to acetylene, this is a chemical or a metabolite that is native to this organism. But in a wild type strain, you can hardly detect this compound because it's always a part of the metabolism of the cell. And then the next slide is another example with the same bacterium. Here we grow the bacterium on methanol. So this one has a extended substrate range when it comes to C1 compounds. And the target product here is malate. And again, if you look at the panel on the right side, you can have focus, yes, around the arrow that you are showing there. So malate is actually kind of a part of the TCA cyclus, and it can be derived from oxaloacetate, which is above there. And the interconversion of oxaloacetate and malate is catalyzed by an enzyme named malate dehydrogenase. And there are different types. They can use different cofactors. We have one that is called NDPH dependent and one that is called chionin dependent. And in this bacterium that we work with, it has a chionin dependent malate dehydrogenase, which is more or less irreversible, which means that it can convert malate to oxaloacetate and not the other way around. And this we want to change because our goal here is to have production of malate. So what was done here was to remove the native chionin-dependent malate dehydrogenase and then replace it by overexpression of a heterologously recruited NADP-dependent malate dehydrogenase. And in this way, you could change the direction of carbon flow between these two metabolites so that you can have 0.32 grams per liter of malate produced from methanol. Yes, and then you can go to the next slide. Yeah, this is more like a scheme showing an overview about how we work. I think this was a little bit similar, like Bettina showed. You have a microbial cell factor, you have an organism you work with, you do genetic modifications, you test it out in small scale. In particular, you're looking at production, of course, but also sometimes looking at growth properties or other behaviors of your cells. If you have a promising strain, you move further to fermenters to try to test out your strain under more industrial relevant conditions. Otherwise, you feel that the strain has a little bit failed, you do measurement, you do analysis, design, and then you go back and then you do new genetic modifications. So this is kind of the cycle that you work with when you work with a combination of synthetic biology and systems biology. And then next slide. Yeah, so I think that these were a few glimpses about what we are doing in our project. And just to put up a few perspectives, what I see is that there is an increased interest for one-carbon feedstocks in industrial biotechnology. We started with this some years ago. Today, there are no commercial processes using one-carbon compounds. So there is a way to go, but still there is a lot of interest in the research also from industry because they see this as one potential way to have a more sustainable industrial biotechnology. Methylotrophic bacteria, of course, are then the main target. There are some challenges with them because you have to look into the metabolic capacities depending on what you want to produce. Genetic tools, there are this is a struggle. We have worked with our bacteria for some years. You have to invest a lot of research to establish the tools to do what you want. And also there might be challenges related to upscaling, but it is improving. So I would like also here to mention that there is one alternative approach which has been given the name synthetic metallotrophy, which also has gained a lot of scientific interest. An idea here is to engineer metallotrophy into well-established bacterial hosts that are already used in industrial biotechnology, such as Escherichia coli, Corynebacterium glutamicum, Bacillus subtilis, and so on. A lot has been done, but it's very challenging. Metallotrophy is complexly regulated. But these two approaches, use the native heterotrophs and synthetic metallotrophy, I think are in parallel now going in order to enable C1 compounds for industrial biotechnology. And I think on my next slide, I just say thank you. Yes, so that was what I was aiming to say. Thank you very much.
Thank you very much, Professor Baltaset. I had some questions myself, but you answered them all. There is like one, I have to stop sharing the screen so I can check actually the chat. Let me see, chat. Okay, have you thought about? Okay, there is a question here. It says, what are the approaches to overcome decreased solubility of CO2 in the medium as the temperatures increase for the cultivation of thermophilic?
Yeah, I think that at 50 degrees, it's still not a big issue. Maybe Bettina also can comment on this, but at 50 degrees, this is still not a bottleneck as I see it from our side. But Bettina, you can maybe comment since you work at higher temperatures.
Yeah, but I think you are right. So I think this is quite reasonable. Yeah.
Okay. There is another question here. It says, how would one increase the capture of CO2 to use it for the feedstock production?
Yeah, so it's important to say that, I mean, there are microorganisms that, the auxotrophic ones, that where CO2 is the main carbon source. That is not the case in our bacterium. It's more like that some of the roots that we work with has this fixation of carbon dioxide. But I mean, if you go to photosynthetic bacteria or photosynthetic microalgae, then you really use CO2 as the true carbon source. So in our case, I think it's mainly methanol that is the carbon source, and then there is a certain fixation of carbon dioxide. Yes.
Okay. I have a question here. So I was reading one of your papers that methanotrophs are hard to manipulate, like for genetic engineering. Why? What is the reason of that difficulty?
It is hard to say what is the reason, because if you look at the two microorganisms, bacteria we use in our project, the Methylobacillus, it turned out to be much easier to develop genetic tools for. When it comes to Bacillus methanolicus, it was like we tested almost all that existed on replicons and plasmids, and then it's also hard to deliver the transformation process. It has something to do with the cell wall. So, I mean, I have worked with the different bacteria, Streptomyces and so on, and some of them just turns out to be very, very difficult to work with genetically. So for us, it's been like a 20-year struggle where we gradually have built up the genetic tools. Now we have different replicons, we have compatible plasmids, different promoters that are adjustable. We have CRISPR-Cas interference techniques, knockout techniques, but this has not come easy. It has been, a continuous development. So, and when it comes to methanotrophs, I know that not many of them are very well developed. Some of them people have worked with. Researchers don't like too much to spend all their efforts into establishment of genetic tools because it's not very high credit. It's something you have to do in order to move on. So, but it's hard to say. But this is a challenge always when you see a bacterium that has some innate properties that you like. Yeah, what about the genetic tools? And this is sometimes a huge challenge.
Okay. I was just curious. So to introduce, let's say, plasmids into this thermophilic or methanotrophic bacteria, you can use like electroporation or just
Yeah.
like heat shock, and it works.
Yeah, so
Okay.
we use electroporation, a very special method, very delicate. We can, at the best, have if we optimize everything, we can have like 100 transformants,
Okay.
colonies. We also, after a while, established conjugation, which has been very valuable for us because conjugation has some, and this was also one reason why we could establish CRISPR-Cas technology. Electroporation has some limitations, but yeah, more or less everything has been kind of resistant when it comes to this organism. But now that we have it, it's a very nice bacterium to work with. But yeah.
Yeah. We have one last question here that I have. It says, how efficient is native methanol dehydrogenase, and what cofactor it uses? Can these be improved?
Yeah, very good question. So I can answer for Bacillus methanolicus has 3 methanol dehydrogenases. We don't know exactly what is the role of all of them, but they are all biochemically characterized. The funny thing is that when you move these enzymes to other bacteria, this seems to be not very efficient in converting methanol into formaldehyde, but for sure they are more than sufficient in the native host. The cofactor is NAD for our enzyme, which is a soluble methanol dehydrogenase, but this is different from other metallotrophic bacteria. Yeah, but the challenge with metallotrophy is that methanol is converted to formaldehyde, which is toxic to any cells. And this further steps from formaldehyde has to be very carefully regulated. So methanol dehydrogenase has to be super efficient to take the methanol into formaldehyde, and then we see it with Bacillus methanolicus that if you give it a little bit too much methanol, it dies. And the reason is that it gets rapid accumulation of formaldehyde, which kills the cells. And I think this is one of the big challenges with synthetic metallotrophy. You can recruit the genes, and on paper, everything should be there. But the problem is that you cannot fine-tune the regulation so that you have efficient conversion of methanol to formaldehyde, and then formaldehyde further disseminated or assimilated by the cells. Yeah.
Yeah, it seems like I feel like working with those organisms at high temperatures, I feel like it's a challenge, and they're not like the common that you use. You have to kind of find a way to tweak them and make them happy.
Yes.
Yeah, so it's now 10:07. I'm so sorry, I'm past 7 minutes. So thank you very much, Professor Sievers and Professor Brauteset, for accepting my invitation to present your work. It's fantastic. I really want to go back to lab. Like, I want to take that PhD position. Like, I want to go. And thank you everyone for your questions and for joining us today. And thank you very much. Bye.
Thank you, Evelyn.
Bye.
See you. Bye-bye.